Welcome back to Medical Neuroscience, and welcome to this tutorial on the modification of neural circuits in early neonatal life. Our core concepts that pertain to this session are, first of all, that life experience affects the function and the structure of the nervous system. This is really the whole point of this discussion today, is to talk about how sensory motor experience changes the nervous system. Well, for me anyway, this topic certainly pertains to one of our core concepts in neuroscience, and that is that our human brain is what endows us with a natural curiosity to understand how the world works. And this has been the case for myself. What I'll be speaking to you about today pertains in part to some research work that my colleagues and myself have undertaken over the last decade or so. So for me, this certainly is a topic that is born from my own sense of curiosity. And of course, I have my brain to thank for that. And then lastly, this topic that we will discuss today is going to be largely one that will relate to primary research discovery. And it's certainly my hope as a neuroscientist that fundamental discoveries will continue to promote healthy living and the treatment of disease. And one of the really fun things about neuroscience discovery is that often basic neuroscience discovery can lead to innovation in the treatment of the human condition in ways that we might not expect as basic neuroscientists. So I think that one is never quite sure where significant application and translation will come from as long as we continue to pursue our curiosity and to continue to pursue basic discovery in whatever domain of science you may be involved in. And for me, that's especially exciting to be involved in the ever-expanding field of neuroscience. Our learning objectives today are first that I want you to be able to discuss the significance of genetic specification, self-organization, and sensory motor experience for the construction of neural circuits in the cerebral cortex. I want you to be able to discuss the significance of experience for the plasticity of neural circuits during what we'll call critical periods of postnatal development. And lastly, I want you to be able to return to a concept that we discussed way back in Unit 2 of the course, and that is Hebb's postulate. And I want you to be able to discuss the relevance of Hebb's postulate for understanding developmental plasticity. Well, I'd like to start our discussion today, as I have the last couple of sessions, and that is by providing a broad overview about the major forces that are shaping brain development. Now, as I think you've been able to appreciate so far, these forces interact with one another, and they do so with one theme perhaps dominating at a different stage of development more so than the others. Obviously, sensory motor experience doesn't become relevant until neural networks are functioning and begin to be modulated by the interaction of the developing child with the outside world. So sensory motor experience is an influence that is emerging in brain development relatively late in the process. Nevertheless, it is an important factor. And what we want to consider today is really, when does sensory motor experience become a major force in brain development, and how does that force interact with these other two, genetic specification and self-organization? Well, just to refresh your understanding of these concepts, genetic specification refers to lineage-derived factors that reflect the spatial and temporal patterns of gene expression. So perhaps it's convenient to think of this as nature, that is, a reflection of the genetic instructions that each cell in the nervous system inherited from its parent cell. Well, self-organization is a relatively underappreciated concept, I would suggest. Self-organization reflects the dynamical behavior of neural systems once synaptic connectivity is established and becomes functional. And self-organization reflects cell-to-cell interactions that are mediated in an activity-dependent fashion. And I'll emphasize activity to describe self-organization rather than experience, because much of the activity in the nervous system is generated endogenously. That is, it is ongoing. It doesn't require external drive to generate that activity. And this endogenous activity provides the substratum, if you will, for self-organization to play itself out. And we'll see a powerful example of that today. Well, sensory motor experience becomes possible relatively later in the story of early brain development as neural systems begin to transduce electrical and chemical and mechanical information that might be present as the developing fetus and developing infant begin to interact with the external environment. And these interactions then produce electrical signals that are driven from external sources and have the capacity then to modulate these ongoing patterns of activity. And all of these factors, genetic specification, experience, self-organization, they interact in complex ways that we still don't quite understand, and the result is the coordinated development of a nervous system that becomes progressively tuned in order to experience the world and respond with appropriate behavior. And this remains much of the challenge of modern neuroscience to understand exactly how this happens. So what I would like to do for much of this tutorial today is to really focus on what we might call nurture. That is sensory motor experience, the environmental interactions that begin to shape the epigenetics that can modulate genetic specification, and also those modulations that potentially can have a role to play in shaping the outcome of self-organization. So I want us to consider the contribution of sensory motor experience to brain development by focusing in on one key sensory system that has served as one of the principal models for understanding these forces in early brain development. And that model system is the visual system. And I'm going to break this down into really two principal parts. One part will pertain to those properties that are present as the thalamus first supplies inputs to the middle layers of the cerebral cortex. So in this case, we're talking about inputs from the lateral geniculate nucleus that are terminating in cortical layer 4. Now, you'll recall, as we've talked about now since the beginning of Unit 3, that one of the principal architectural features of the cerebral cortex is a columnar circuit that repeats many, many times across a cortical area. And we think that that columnar circuit serves at least three important functions that I've suggested you remember by recalling the ACC of the cortical microcircuit. The first function that's performed by this circuitry is amplification, and that's principally amplification of this thalamic input. So these inputs are processed among small cells that we call stellate cells in cortical layer 4, and from there the information is passed on up into the upper layers of the cortex, and from there down to lower layers of the cortex. So there's sort of a recurrent columnar circuit that amplifies the input that is received in the middle layers of the cortex. We'll go on and talk about the computation of new cortical properties in the next part of this tutorial. So what I'd like to talk about in this first part of the tutorial are the forces that shape the initial topographic mapping of this thalamic input into cortical layer 4. And just to give you a sense of what the outcome of a long series of experiments suggest is that there's an early phase to the development of this system that some have called a pre-critical phase, to emphasize the idea that this phase establishes the basic mapping function that allows the thalamus to map into layer 4 of the cortex, and it's pre-critical in the sense that it doesn't seem to require visual experience in this case, since we will be talking about the visual cortex. Now, this implies then that there must be a later phase that we'll call a critical phase or critical period, where sensory experience, visual experience, can indeed alter the structure and the function of the circuits that are initially established as the thalamus maps into layer 4. Well, as I mentioned, we're going to be talking about the visual cortex. Just a little bit more background to remind you of where we're going with this. So the visual cortex that we have in mind really is the primary visual cortex, which is what we find in the banks of the calcarine sulcus here. This is a beautiful image supplied by my colleagues in our field of visual neuroscience, Dr. Daniel Adams and Jonathan Horton. This part of the brain is exquisitely organized in so many different dimensions, unfortunately beyond the scope of our consideration in this course. But it has captured the imagination and the attention of now at least 3 generations of neuroscientists who've been studying this part of the brain. It is undoubtedly the best understood region of the cerebral cortex, so it serves us well as a paradigm for understanding some of the principal forces that are shaping the development of the brain more generally. Well, this cortex, as you know, is topographically organized. The fovea representation is found at the occipital pole of the hemisphere, and as one progresses from the posterior towards the anterior aspect of this calcarine sulcus cortex, one moves from the central region of the visual field out towards more peripheral regions of the visual field. And this beautiful color code as visual field is related to visual cortex reminds us of this orderly progression of visual topography. It also highlights the fact that what's most important to us in vision is represented by nearly half of the visual cortex, and that is the central 10 degrees or so of the visual world. So it's really the representation of the fovea, and surrounding the fovea, the macula, and then the nearby regions of the retina that account for much of the detail that captures our visual interest. And this is represented by circuitry that spans about half of the visual cortex. It's not to say that what happens in the periphery isn't still relevant for visually guided behavior. Of course it is. But it doesn't require as much circuitry to analyze the details of form and color and location as what we find in the representation of our macular region of the retinas. Now I'll just remind you of what this region of the brain actually looks like histologically speaking. Here's our calcarine sulcus that we see in a histological slide that's stained for the presence of myelin. And on the superior bank of this calcarine sulcus we have the cuneus gyrus, which is going to be representing the contralateral inferior quadrant of visual space, whereas on the inferior bank we have the lingual gyrus, which is representing the contralateral superior quadrant of visual space. Now, this part of the brain is often called the striate cortex because of this densely myelinated stripe of cortex that we see that runs right through the middle layers. So while this stripe is not precisely layer 4, it's found within layer 4 of the visual cortex. So the first part of the story that I'd like to tell you today about the formation of topographic maps and how sensory experience can alter the structure and function of those maps really pertains to establishing the initial connection from the thalamus to this region that's recognizable by this heavily myelinated stripe in the visual cortex. Now you may recall that one interesting feature of this middle layer of the visual cortex that was discovered back in the mid-60s to late 70s is the system of ocular dominance columns that we find in the middle layer of the visual cortex. So let me remind you about ocular dominance. As you know, the two eyes are sending signals into the same lateral geniculate nucleus, but as you'll recall, those signals are not yet combined at the level of the thalamus. Rather, the two eyes project to separate layers of cells within the lateral geniculate nucleus. The contralateral eye projects to layers 1, 4, and 6, whereas the ipsilateral eye projects to layers 2, 3, and 5. It's not yet until these circuits are amplified at the level of the visual cortex do the inputs that are driven from the two eyes first become combined. And in fact, even in cortical layer 4, there's still segregation of these inputs. So as the thalamus projects into cortical layer 4, there is initially a termination of afferents that segregate very quickly into complementary zones, such that one can progress across the visual cortex within layer 4 and encounter these alternating bands or alternating columns, if we think in the radial dimension, that are driven primarily, if not exclusively, just by one eye. Now, once we get above cortical layer 4, the inputs do become mixed. And so this means that stereopsis is first possible in the upper layers of the visual cortex, where information from the two eyes begins to converge on the same cells for the first time in the visual pathway. Nevertheless, even in the upper layers of the visual cortex, there's still a bias towards information that's being driven by one eye or the other. Well, this fact was not lost on David Hubel and Torsten Wiesel as they discovered ocular dominance columns together with their collaborators such as Simon LeVay and Carla Schatz. And what these individuals recognized is that this system of ocular dominance columns in the visual cortex presents a wonderful opportunity to begin to design experiments to look at the role of experience in early life. Well, what was especially powerful about this system is that not only could this system be probed physiologically with microelectrode recordings, and one could assess the degree to which one eye or the other was responsible for driving the action potentials recorded from a neuron in any one of these ocular dominance columns, but there's also an anatomical correlate that could be visualized through various means. And I'd like to show you one other image from my friends, Dr. Horton and Dr. Adams. And this is now one of my very favorite images in the entire field of neuroscience. What we're looking at here is a photo montage constructed from the human visual cortex. And this is tissue that has been prepared and stained to demonstrate the presence of a oxidative enzymes that's found in mitochondria, cytochrome oxidase. And so the intense dark brown regions are those regions that have high levels of cytochrome oxidase activity, and the more pale regions are those with low levels of cytochrome oxidase activity. So the intensity of the brown tells you something about how metabolically active the individual locations in this cortex were in life. Now, this visual cortex was derived from an individual who passed away after having one eye removed. And as a consequence, the ocular dominance columns or stripes in the visual cortex that were driven by the eye that was removed had down-regulated their oxidative metabolism. And as a consequence, the columns that were driven by that eye have now become much less metabolically active. And hence we can reveal for the very first time the complete pattern of ocular dominance columns in the human visual cortex. Now, take my word for it, this was a Herculean effort. Very few histologists are skilled enough to have produced an image such as this. What Dr. Adams and Dr. Horton and their staff were able to accomplish here is truly a remarkable bit of anatomy and histology. What they were able to do is to gently unfold the cortex of the occipital lobe in the upper and lower banks of the calcarine sulcus, to flatten out that cortex and then to produce sections through it that then could be stained to demonstrate this metabolic activity. And what we see is truly a remarkable picture of the structure of the human visual cortex. I hope you're struck, as I am, by really the beauty of this image. So what we're looking at is a representation of visual cortex with the posterior pole of the hemisphere here to the left side of the image, the anterior side of the calcarine sulcus cortex to the right, with the superior aspect, the cuneus gyrus above, and the inferior aspect, the lingual gyrus below. But really the main feature for you to recognize is this beautiful pattern of stripes and columns that we see. So these are indeed our ocular dominance columns, or ocular dominance stripes, as we might recognize them here in this broad view of the visual cortex. So these dark regions are those that are driven by the eye that remains, and the intervening pale zones are those that were driven by the eye that is now removed, and hence they have down-regulated their enzymatic activity.